Hi, I'm Ashira Blazer, an assistant professor of rheumatology at Hospital for Special Surgery. Today, I'm going to be talking to you about my work recently published in Frontiers in Genetics entitled APOL1 variant expressing endothelial cells exhibit autophagic dysfunction and mitochondrial stress. So, what is APOL1 and why are we discussing it today? So, APOL1 is encoded on chromosome 22. And when looking at the amino acid structure of the reference or G0 allele, the G1 variant encodes two amino acid substitutions, and the G2 allele encodes a two amino acid truncation. Now, these variant alleles are very common in African ancestry populations, and about 13% of African Americans carry the high risk genotype, meaning carrying APOL1 variants, either G1 or G2, in any form. Now, this is disease associated, so increases the risk of chronic kidney disease due to FSGS, HIV associated nephropathy, or lupus nephritis, among others, and also increases the risk of some vascular disease, such as microvascular stroke and peripheral vascular disease. Now, why are these so common in African ancestry genomes? And it turns out that this story is somewhat similar to the sickle cell story, where carrying these variants in any form decreases the risk of dying from infection, in this case, African trypanosomiasis or African sleeping sickness, but when carried in homozygous form, increases the risk of disease. So APOL1 is expressed by numerous cell types, including hepatocytes, which can secrete APOL1. And when this happens, as a lipoprotein, APOL1 travels along with these HDL3 particles as the trypanolytic factor. When someone becomes infected by blood parasite, Trypanosoma brucei, these parasites will start to take in fatty materials in the nascent environment, including these HDL3 particles. Now, these will be home to the lysosome, where under this acidic pH, APOL1 will change conformations and sort of come off of this HDL3 particles. And there's a pore-forming domain, which is able to traverse phospholipid bilayers. This can insert in the lysosome. And this means that this trypanosome has its digestive enzymes leached out, and this causes trypanosomal lysis. Now, in areas where trypanosomiasis is endemic, there's been this evolutionary arms race. So APOL1, the native G0 APOL1, could lyse trypanosomes, and trypanosomes mutated such that they could bind the C terminus of APOL1 and neutralize this protein. So in turn, these variants arose where the C terminus was not so easily bound, and so this restored the trypanolytic activity of APOL1. Now, when looking into C at this protein, you've got multiple domains. So you've got this signaling peptide, also secretory peptide, which is cleaved in most cells. You've got the pore-forming domain, which traverses phospholipid bilayers by way of this membrane addressing domain. Now, within the pore-forming domain, there's this BH3 domain, which allows APOL1 to participate in autophagy, which is a cellular maintenance process. You've got this SRA domain, or the serum resistance associated, which is where these variants lie. Now, compared to the protein that's encoded by the G0 allele, there are these very tightly wound alpha helices. So these helices sit together and give the protein this structural stability. Now, the G1 and G2 alleles encode proteins that have decreased structural stability at these terminal coiled helices. So these non-synonymous amino acid substitutions are not able to form hydrogen bonds. So you get separation of these coiled helices. And this G2 allele has this telescoping out due to that truncation. And so this is associated with APOL1 being less able to be bound by trypanosomiasis, but also being less easily bound by its intracellular binding partners, namely BCL2 family proteins. Now, let's talk a little bit more about APOL1's autophagy function. Let's pretend like this slide is the intracellular environment. So under conditions of stress, such as low oxygen tension, low ATP, high AMP, or some sort of inflammatory stress, The cell is going to initiate autophagy because autophagy is a maintenance process. So this is how cells take long-lived proteins or defunct organelles, engulf them in autophagosomes, and deliver them to the lysosome for degradation. And this can be harnessed to produce energy under times of stress. Now, one thing that happens is that BH3 only proteins, of which APOL1 is one, start to become expressed. And so these proteins like APOL1 can compete for the binding of anti-death BCL2 family proteins. And when bound, you competitively knock off Beclin-1, which participates in propagation of the autophagosome. Now, the end step of autophagy is autophagic flux, where these autophagosomes are delivered to the lysosome and the contents are degraded. Imagine if you have variant APOL1 that's not so easily bound by these BCL2 family proteins. Well, now you've got free APOL1, which can home to one's own acidic organelles, namely the lysosome and the mitochondria. And this means that you have disruption in lysosomes and decreased autophagic flux because you cannot digest these autophagosomes. Also, a decrease in mitochondrial oxygen consumption and therefore ATP production. And we think that this is related to the toxicity model of APOL1. Now, why are we studying this in a lupus relevant model? Well, it turns out that inflammatory processes lead to increased APOL1 expression. So in lupus, you've got ligation of toll-like receptors. This leads to downstream activation of transcription factors, which bind to the promoter of APOL1. This leads to increased expression of APOL1 and increased accumulation of the protein intracellularly in cell types such as endothelial cells. Now, you also have a type 1 and type 2 interferon response, which comes back and acts on the cell to produce even more APOL1. So inflammatory or infectious scenarios may be ones that increase the consequences of carrying these variants because it increases the expression and accumulation of the toxic protein within certain cell types. So we created these HUVEC cultures representing each of the APOL1 genotypes. And we either left them untreated or treated them with lupus-relevant stimuli, including interferon alpha, interferon gamma, and TNF alpha. And under all of these conditions, we got increased expression of APOL1 and increased APOL1 messenger RNA within these endothelial cells. This happened most robustly when treating with interferon gamma, and so we use this as a reagent for the rest of the experiments. On the protein level, regardless of genotype, so these are HUVECs that carry 0 copies of the risk variant, 1 copy of the risk variant, 2 copies of the risk variant, we got an increase of APOL1 expression compared to tubulin when treating with interferon gamma. We also added hydroxychloroquine, which is an autophagic flux inhibitor. So the next thing we did was take a look at mitochondrial oxygen consumption. Now, we did this with a functional assay called the seahorse assay. And so what this does is it takes plated cells and it measures the basal mitochondrial oxygen consumption. Then we give oligomycin, which is an ATP synthase inhibitor. So the schematic on the bottom represents the inner mitochondrial membrane. So you can't have electrons go through ATP synthase. And so you should have the non-synthase oxygen consumption. The next thing we give is FCCP, which is an uncoupling agent. And so what this does is it allows electrons to be uninhibited in their ability to traverse the inner mitochondrial membrane. And so this asks, if we allow these mitochondria to consume as much oxygen as possible, what's the maximum capacity of oxygen consumption? The next thing we give is rotenone, which is a complex 1 inhibitor, and this shuts down the entire electron transport chain. And so this is a non-mitochondrial oxygen consumption. So healthy cells should have relatively higher basal oxygen consumption, maximum oxygen consumption, and lower non-synthase and non-mitochondrial oxygen consumption. So here are representative tracings from cells representing either 0 risk variant carrying HUVECs, 1 risk variant carrying HUVECs in the purple, 2 risk variant in the gold. And these were either left untreated in the lighter bars or interferon gamma treated in the darker bars. And what we can see is that under both conditions, the 0 risk variant carrying HUVECs had the highest basal oxygen consumption and the highest maximum oxygen consumption. Now, in untreated HUVECs that carried one copy of the risk variant, you had comparable basal and maximum oxygen consumption, but when you gave interferon to induce APOL1 expression, you got this drop down of both. In double risk variant carriers, there was this constitutively active decreased basal and maximum oxygen consumption. The next thing we wanted to do was take a look at mitochondrial morphology. And the reason for this is that mitochondria should actually form these nice networked branches. So mitochondria that have a nice membrane potential can fuse with each other and make these conglomerates. When mitochondria are less energetic, they don't have such a high membrane potential, they are unable to fuse and you have these individual fission forms. Now, these can be engulfed in autophagosomes and degraded via a pathway called mitophagy, which is an analog of autophagy. So more of these fission forms or small piecemeal mitochondria, less energetic mitochondria. In the panels to the right, I'm showing representative HUVECs from 0 variant carrying HUVECs, 1 variant carrying or heterozygous, 2 variant carrying or homozygous for APOL1. Now, I'm staining here mitochondria in the green mitotracker and lysosomes in the red lysotracker. So you can see in these 3 HUVECs that carry no copies of risk variant, the mitochondria are nice and lacy, these spaghetti-like conglomerates. So many fusion forms. You also have relative abundance of lysosomes. When you have 1 copy of the risk variant, you have more of these fission forms and fewer lysosomes. And in the double variant carrying HUVECs, you have many of these fission forms and very few lysosomes, suggesting disruption in these 2 organelles. So taking a closer look at the lysosome, we stained these HUVECs with lysotracker. So the rows here, 0 variant carrying HUVECs, 1 variant carrying, 2 variant carrying, so each of these boxes are a representative HUVEC, and we've stained with lysotracker. So the more red hot these cells are, the more lysosomes you're seeing. So again, there's a stepwise decrease in the lysosomal compartment. And when we give hydroxychloroquine, which neutralizes the pH of lysosomes and therefore decreases turnover of lysosomes, you can see that this comes out even more. So you've got many lysosomes in the 0 risk variant carrying HUVECs, stepwise decrease with each additional variant. Now this is characterized, this is quantified in over 15,000 cells by integrative density of the lysotracker staining in 0 risk variant carrying, 1 risk variant, 2 risk variant carrying HUVECs. Now, the next thing we did was treat with interferon gamma because this should induce autophagy and lead to relative turnover of lysosomes. And you can see that you do have a stepwise decrease with giving interferon, but that this decrease is most apparent in the double variant carriers, which have very few lysosomes under that condition. Okay. So if you have a decrease in the lysosomal space, so fewer lysosomes, you should have this antecedent buildup of autophagosomes because you're disrupting autophagic flux. So again, we have the rows, 0 risk variant carrying, 1 risk variant carrying, 2 risk variant carrying HUVECs. So each of these, you see the nuclei are stained with DAPI in blue. There's a red counter stain. And we're also staining P62. So P62 is an autophagosome shuttle protein. So the more of these green puncti you're seeing, the more autophagosomes that you're seeing. So the untreated state is the top row, interferon treated state is the middle, interferon plus hydroxychloroquine is at the bottom. And here we gave hydroxychloroquine because it neutralizes the lysosome pH and decreases lysosomal function. So we should induce an autophagic flux defect under these conditions. Now this is quantified in the graph here on the right with the log ogophagosome count per cell here on the y-axis. And you can see the different conditions across the genotypes. Now, in each of these, you should have an autophagic flux defect with a hydroxychloroquine treated state. And so you should have many autophagosomes here compared to the untreated state where you have fewer. So this gradient with interferon inducing autophagy and the hydroxychloroquine blocking autophagy should be pretty steep, as you see in the 0 variant carrying cells. The gradient is less steep with 1 risk variant copy, suggesting that there's some autophagic flux defect at baseline, and almost flat in the double variant carrying cells, suggesting that there's this constitutively active autophagic flux defect. So, what are the translational implications here? APOL1 is expressed in endothelial cells where the variant protein may disrupt autophagic flux as well as mitochondrial health. Interferons increase APOL1 expression and variant-associated cytotoxicity. So this may be an underpinning for the heightened associations that we see between carrying the high-risk genotype and kidney disease in states like lupus nephritis or HIV. Also recently, COVID-associated nephropathy has been associated with carrying these variants. Now, I want to take a moment to talk about this paper and the journey that I went on with it. So I first submitted this paper about 7 years ago, and here are all the journals that I've tried. There have been many, many submissions and resubmissions and new experiments trying to get this published. And along the way, I have to say that I struggled, right? So the imposter syndrome that I experienced, the difficulty with keeping going, envisioning myself as a scientist or a functional genomics expert was really, really challenging. But despite the fact I kept going and kept going with the help of all of my mentors and all of my co-authors, and this eventually got published in Frontiers in Genetics. So I'm very proud of this work. I hope that it was informative to you, and I hope that you enjoy the rest of your day.